my students, don't worry about putting down your student ID number. Just write down your name legibly and that you're a Squires student. Okay, so my pleasure to introduce Dr. William Self from UCF. He did a lot of his work at the National Institute of Health, and then about 16 years ago, moved to UCF where he runs an awesome lab. Really excited to hear about his work. If you have any questions during the talk, please just raise your hand and he'll call on you. Okay. Thank you.
Thank you, Dr. Squires, for inviting me. I think I'll try and figure this out. Okay, so thanks for the invitation. And so yeah, you can interrupt me at any point if you have a question. I always, when Dr. Squires asked me for a title, I remembered back to the very first talk I ever went to. And it was at a microbiology meeting in New Orleans, and I think it was '96, and the big meeting with about 10,000 scientists. And the very first talk I went to, the title of the talk was "How Anaerobes Make a Living," and it was by a scientist named Gerhard Gottschalk, who had written this book called Bacterial Metabolism years before. And I went to Gottschalk talk because he was like world famous for that. And then when I got to the talk, he was pretty old at the time, and he said, "Well, I'm changing my talk to how to make a living studying anaerobes," and it was all about his career. But I still remember that talk, and he did talk a little bit about anaerobes. But one of the things that my lab has done over the last 16 years, and I'll talk a little bit about my mentor, Terry Statman, is studying how Clostridia or how anaerobes make a living. And in this case, we've studied some aspects of Clostridium difficile, which is a pathogen, which is a nosocomial pathogen that you find, unfortunately, in the clinics and in the hospitals. So my background is in microbiology. I did my undergraduate and my doctoral work at UF. I did do a 5-year postdoc up at the NIH in Dr. Terry Statman's lab, so I'll probably talk a little bit about her discoveries as well. And then I've been at UCF for 16 years. I also oversee our undergraduate programs now in the Burnett School of Biomedical Sciences. Since the time I came to UCF, we've grown from a department to a college, and then we were brought into the College of Medicine when it was started, so about 10 years ago. We have about 3,500 undergraduate students in our programs at UCF, and some of you actually be coming over to UCF at some point in your career. My interests still lie in the areas of bacteriophysiology and metabolism, especially the use of metals and metalloids in enzymes in bacterial model systems. I'm not going to talk at all about it today, but my graduate work was on the transcriptional regulation of molybdenum-containing enzymes in E. coli. And in fact, it's a lot, the studies that we did and a lot of other groups did in E. coli sort of relate to some of the work that's going on now in C. diff and how we're understanding C. diff a lot better. I've gotten a couple of other complete areas that I won't talk about. We have worked on the area of toxicology of arsenic and selenium. The only thing I'll mention on this is that that arsenic and selenium are actually both very toxic metalloids. And so you hear about arsenic as a poison. Arsenic is a poison. Selenium is just as much of a poison as arsenic. And there was some work many, many decades ago on this mutual sparing effect, and we did some work at the molecular level on that a number of years ago, but that's not part of this talk. And I've also expanded out with collaborations in material science and engineering and studied some interesting cerium oxide catalysts, and we published a number of papers on that in the last 16 years, probably about the last 12 years. I'm not going to talk about that as well, but really some interesting collaborative work that came out of conversation at a breakfast, which is an interesting way to start a big collaboration. We're still working in this area, but sort of going to go back to my roots in terms of bacteriophysiology today. So a lot of folks, including Dr. Squires' mentor, Dr. Marla Berry, has worked on selenium in biology. Again, it is a poison, it is toxic. Let me see if I can figure out this pointer. It's genotoxic and it's carcinogenic. One of the earliest studies in selenium toxicity was in California, in a place called the Kesterson Reservoir, where there was an agricultural reservoir that ended up very, very high in selenium levels, and it ended up killing off a lot of the plant life and birds, and that was sort of so also from what I understand from my friends in biology, it's taught as a sort of an ecological disaster in some of their coursework. There was a correlation between something called white muscle disease and low selenium levels back in the 1960s. And there was some evidence that selenium was a nutrient in animals early on and in USDA-funded studies. And then later, the first report of anything with respect to E. coli, with bacterial systems, was a paper in 1971 by a guy named Bob Lester, and he actually showed that the addition of selenium in the form of selenite helped to produce hydrogen, helped in the production of hydrogen by E. coli. And what's interesting behind this is that this was in fact later shown to be, and was developed in Terry's lab, a selenoprotein called formate dehydrogenase H. And yet they didn't know what the selenite was doing in that paper in 1971. But it was right around the early 70s when some of the evidence for the use of selenium in biology, that's sort of the point of this slide, is that prior to the 1960s and 70s, selenium is only seen as a poison like arsenic. In other words, there was no known biological function of selenium as an element, yet some of these early studies, and this was the first bacterial study. So my mentor, Terry Statman, had done her doctoral research on an organism that she isolated from the San Francisco Bay mud called Clostridium sticklandii. So she isolated that during her PhD work. Her and her husband actually moved to the NIH in the 1950s. She had studied an enzyme system in this organism called glycine reductase for a number of years while she was at the NIH. And they worked in a building called Building 3. It was an older building even at the NIH back then. And they got a new still. So they had been using one type of water for the fermenter, and they had a new still put into the building where they were basically getting cleaner water, and it was removing some of the contaminants. So Terry told me the story, and many times. She was working on the fermenter, she grew up her organism, and with the still, with the water, it didn't grow that well. So it went from growing pretty well to where it didn't grow that well. So she knew that there was some contaminant in the water that was helping in growth, but she didn't know what it was. So she basically took everything off the inorganic shelf, inorganic chemistry shelf, and added a small amount to each one of those elements in small batch cultures to find out which element was helping her Clostridium grow in a test tube. And it turns out that sodium selenite increased both the growth of the organism and it increased the enzyme activity. So she started adding it to her cultures. And she had been studying this glycine reductase for probably about 15 to 20 years prior to this, always isolating it from the same organism, Clostridium sticklandii. But now she had this element that she was noticing helped grow the organism. And the interesting thing about this is there's probably about 5,000 proteins in any bacteria. So 5,000 genes encoding 5,000 proteins, right? She's studying one gene out of 5,000, and then she has a problem growing it in the test tube, right? What are the chances that that has anything to do with her protein, given the odds, right? 1 in 5,000 odds. Long story short, the selenium atom was present not only in her protein, it was present in the active site of the protein, and they were able to show that with radioactivity, a couple of years after she actually discovered it, it was about 71 when she discovered it in the lab, the selenium was actually in the active site of her enzyme. And so that's just one of those things that you can't believe when you think about the chances. So then she purified that protein. One of her postdocs purified that protein, published a paper in 1973 that showed that for every, what we call mole, for every mole of enzyme, you had a mole of selenium. And that was the very first published paper that showed that selenium was present on purpose in a biological molecule, right? So selenium had known to be incorporated into plants and had known to be present in cells for no known purpose. This was the first isolation of this. In fact, that year was a banner year because the incorporation of selenium into formate dehydrogenase from another Clostridium was reported 2 months after this, and the insertion of selenium in glutathione peroxidase by Flowey was reported, I think, about 6 months later. So that year, multiple investigators had published papers saying selenium has a use in biology back in that. And really, her uncovering of this was an accident, right? So she had just gotten a new still. If they never got a new still, I don't know if she'd actually discovered. Someone would have discovered it years later, but certainly this was an interesting way to discover that. So that era is where we went from thinking of selenium as a poison to where people started thinking about selenium as a nutrient, micronutrient. And of course, if you take Centrum, or I take like Costco and generic vitamins, it has selenium in it today. It's not really the focus of the talk, but if you want to ask questions about that, we can certainly talk about it. So this is a pretty busy slide, and I'm not going to go over every aspect of it, but there's various forms of selenium in biology. There's various forms of selenium in the soils and sediments. Selenate and selenite are the most abundant. These are the oxyanions of selenium that you find in the soils and sediments. Selenomethionine, selenocysteine, these are the forms of selenium that are dominant in us. So pound for pound in us, we're made with selenocysteine and specific selenoproteins and also non-specific selenoproteins. Selenomethionine, when you add selenate to plants, you typically get a lot of selenomethionine production. You get a lot of selenomethionine in humans as well. We can use it as a source of selenium. However, the core pathway that has been described by Terry's group, Marla Berry's group, August Beck, all of these groups that studied this early on, is this pathway right here. So this is the specific incorporation of selenium into what we call a selenoprotein. And that always goes through this ironically toxic intermediate, and that's hydrogen selenide. So hydrogen selenide is the precursor of the substrate of an enzyme called selenophosphate synthetase. It's called CELD, or encoded by the CELD gene in E. coli. The CELD uses ATP and converts hydrogen selenide to selenophosphate and AMP in a well-described process now, both in humans and in bacteria. This activated form of selenium gets moved over to the tRNA, and actually you build the selenocysteine amino acid on the tRNA, which is a little bit unusual in biology, rather than just charging the tRNA with a free amino acid. And of course, the selenocysteine was described as what we call the 21st amino acid because it uses a UGA codon and a specific type of elongation factor to incorporate it into the protein. And so this is the sort of canonical synthesis of selenoproteins, primarily being looked at from a bacterial standpoint. The toxicity of selenium correlates with selenide production, and the toxicity of selenium in animals and plants and all of that. There's a number of groups that have worked on that. We've done a little bit of work on that, never really gotten into it. But it's an interesting area that really has still a lot of unanswered questions. So down here, selenium is clearly a double-edged micronutrient. At high levels, it's toxic, just like arsenic, but we need a small amount to go through a specific pathway. And in humans, it's absolutely essential. So we have 25 selenoproteins in humans, and a knockout in most of these, or in the process by which you can make selenoproteins, is embryonic lethal. But in most bacteria, this is expendable. You can delete it, and the bacterium can get along just fine. So that's going to be a little bit of what we talk about in terms of the science today. This is just another way to loop at it. This is some of the work, an old review from August Beck's group. And you have a specific elongation factor protein that binds to a stem loop structure in the mRNA, where you recruit this charged tRNA complex. The ribosome is sitting there and it sees a stop codon, and rather than stopping the protein production, or rather than pausing for too long, if you have substantial amounts of these coenzymes and proteins, then you can make a selenoprotein with reasonable effectiveness and efficiency. This was first described really well, mostly in August Beck's lab in E. coli. So in bacterial systems, there's a number of selenoproteins that have now been defined. Formate dehydrogenase was one of the first. That's the one that was described in E. coli. Some bacteria actually incorporate selenium into their selenophosphate synthetase, which is an interesting thing. We really don't have any idea why they do this or what benefit they get from that. That's an unanswered question. Turns out that two of the proteins that Terry was studying, glycine reductase, selenoprotein A and B, both have selenium. An enzyme that was identified as a selenoprotein back actually when I was a postdoc was D-proline reductase in some Clostridia. We now know that that's present in the organism I'm going to talk about. There are some selenium-dependent hydrogenases, enzymes that either take up hydrogen and use it as an electron donor or produce hydrogen gas, can use selenocysteine. And then there's one critical step in methanogenesis. This is in the archaebacterial, starting to go on timer. There's a selenium-dependent process there. There's a class of enzymes I'm not going to talk about where selenium is actually incorporated into the enzyme, but not in the form of selenocysteine, which we understand somewhat about it, but we haven't fully understood the pathway to this process. We've had a little bit of computational work in this area, and actually I've got a couple of students in the lab right now trying to decipher this pathway in a heterologous system, which is E. coli, but using genes from Enterococcus faecalis. This is not sort of the subject of today's talk. One of the questions we asked when I first came to UCF and was looking for a model system to study is what microbes might require selenium as a micronutrient based on what we know about their physiology. So it turns out there's one paper and one study that clearly shows in what we call defined minimal medium conditions. This is where the minimal defined conditions for an organism to grow. And this is Mike Rother's work back a number of years ago in August Beck's lab, that Treponema denticola absolutely requires selenium to grow. And this is actually an oral pathogen that causes gum disease, significant amount of gum disease in humans. C. diff, we have evidence that suggests selenium is important for growth. We now actually have evidence that says that it's expendable to an extent, but we're trying to wrestle with exactly what that means. Here's some other organisms that we know grow better with selenium, but whether or not they require selenoproteins is still an open question because of the lack of genetic systems. One of these being the organism that Terry isolated out of the San Francisco Bay. But these are not pathogens, clearly, these last three, but obviously botulinum is a foodborne pathogen. So now let's talk a little bit about C. diff, and then we'll give you a little bit of a story in terms of what we've done in the C. diff area, and then open it up for questions. So Clostridium difficile is a gram-positive anaerobic spore-forming bacillus. Transmission is fecal-oral. I write primarily nosocomial because now there's some good evidence that community-acquired disease may be relevant. There are certain guidelines on whether a patient walks in the door into the hospital and is a carrier or has C. diff. There are guidelines as to how long after admission they become nosocomial infection and whether they're bringing it in. So this is an open question in the clinical community, and it's also an issue in the hospitals. We can certainly talk about that in terms of the current burden of C. diff in hospitals. If you've been recently on antibiotics, over 65, if you have an underlying illness, these are the typical risk factors. These are where you get most of the cases of C. diff, but you can get C. diff in very young adults with no real risk factors as well. Hopefully you just can resolve out of a mild diarrhea, but some cases, and possibly an increasing number of cases in the last about 12 years, you can end up in more serious condition where you have pseudomembranous colitis. And about the last good epidemiological study suggested about 38,000 patients in the United States die with C. diff infections per year. But it's very important that you understand the language of that. They died with a C. diff infection. It doesn't mean that C. diff alone killed the patient, but that it's associated with their death. And it's very difficult to say whether it's the causative agent of death or not. But the reporting on that's changed over the last few years as well. Certainly there's a lot of burden in the hospitals. There's a lot of patients that get C. diff, and it sometimes doesn't resolve very well. This is pseudomembranous colitis. It can produce a toxic megacolon. What mediates all this inflammation is two toxins, toxin A and toxin B. It's very well defined that if you take the toxin away from C. difficile by mutation, then it renders the organism unable to cause disease in animals. You can identify and you can actually isolate C. diff from patients that have no toxin, but there's no known threat that is causing disease. There's a binary toxin that's been described that may be contributing to disease as well. These two toxins mediate essentially the vast majority of the clinical symptoms as far as we can tell. And they're sort of also classically used by cell biologists to study Rho and RAS GTPase proteins and their mechanism. But you end up with inflammation, fluid, and mucus secretion, damage to the mucosa. One of the fundamental questions that I have in our lab that we've asked is, why does C. diff make the toxin? So we still don't know from a bacterial physiologist standpoint, someone who's trying to study how C. diff makes living, what is the rationale for toxin production? Because these toxins are 1,100 amino acids, so they're expensive to make. They take a lot of energy to pump out into the culture medium or into into the gut if you're in the host. We still don't understand exactly why they make toxin, because they typically don't, go to the liver. In other words, the target doesn't seem to be that C. diff wants to go to the liver or to necessarily kill the human. So there's got to be some biological reason that they would spend all this carbon, nitrogen, energy to make toxins, and we're still unclear on that. So that's one of those long-term philosophical questions we'd like to answer at the physiological level. So just a little bit more about the toxins. So what I really want to talk about here is there was an emergence of some strains back around early 2000s, and some of the papers call them hypervirulent strains, some of them call quote-unquote epidemic strains. But last year, in 2018, Rob Britton's group published a pretty remarkable study that I point to here that actually shows fairly well that a food additive called trehalose, so this is actually a sugar, and was approved by the FDA as a food additive back earlier than 2001, right around 2001, I've got a timeline, seems to be possibly one of the reasons why these strains expanded out in human population. So there was a rapid expansion of this type of strain across the globe, actually. And it coincides with these strains actually using, or I should say, these strains being able to use this carbon source more effectively and efficiently than other strains. And it coincides with humans eating a whole lot more of this. So the cost of this as a food additive went from very, very high to very low very quickly. I never found out what the technology was that they could make this or isolate this food additive. But the food industry started using it, and C. diff spread around the world. And if you Google that, you'll get a lot of interesting articles on that as well. Here is a couple of slides that show the reports of this quote-unquote epidemic strain across the United States back in 2007 or 5. This is 2005. So these are all we literally watched this as epidemiologists spread across the United States, spread across the world. This was a couple of years later, 2007. This is actually from the 2018 Nature paper, and this is all of the reports of where this group of strains spread across the world. You can actually see here, and here's where the FDA approves trehalose as what they call GRAS. So something you can pretty much add to anything you want to for its chemical properties. So you find it in all kinds of foods in the United States and therefore all around the world as well. So when you look at an outbreak of C. diff, or if you just had been doing surveillance of C. diff around the United States, for example, prior to this, what you would get is a broad distribution of isolates. In other words, there are strains of E. coli that are going around, there's strains of the flu going around. You would get a pretty broad distribution of C. diff isolates when investigators in the clinical labs were looking at what isolates might be there. That diversity seemed to decrease after this, and then the spread of these strains was literally global. An interesting couple of papers were published back in the early 2000s where you could actually see the mutation frequency as the strains spread around the world, because the genomics era occurred right around in here. It was really fascinating from a scientific perspective. Of course, from a human perspective, it's these strains that spread around the world now. And so this is a really, really neat study that Rob's group did last year. So early on in my time at UCF, one of the reasons why we chose C. diff is we wanted to find a model system where we could study the systems that Terry had studied, these amino acid reductases, and determine exactly what their role is in the physiology of these organisms, hoping to cash in and have a model system where we could get some genetics, and that's where we've been fortunate to have collaborators. It turns out that when Terry actually isolated and published an early paper, 1949, on Clostridium sticklandii, the most closely related organism in her study that was not C. sticklandii but was a different organism was actually C. diff. And so it's a very, very closely related strain to the soil organism she was studying. The genome told us that the selenoproteins were there. This is one of the most important reasons, and we still are looking at this today. A number of studies have shown that depending on what C. diff sees in its environment, it adjusts the toxins. So the toxin regulation is now pretty well established. Actually, in the last few months, there have been a number of papers published in the last few years. but the mechanism by which C. diff decides, based on nutrition, to make toxins is still pretty poorly understood. We're getting there. So I'll just get through these. So essentially, the core biology that Terry started studying and what they call the Sticklin reactions, I want to talk about briefly. So humans and E. coli, well, let's just go with E. coli. E. coli is really good at oxidizing sugars. And so E. coli can use a number of sugars as a carbon source. And then E. coli, as an organism, is well established to be an arid, right? So we can use oxygen as an electron acceptor. If it doesn't have oxygen, it can use a lot of different other electron acceptors, nitrate, nitrite, dimethyl sulfoxide, trimethylamine oxide. So there's a number of alternatives for E. coli to respire on. And then if it doesn't have anything to respire on, E. coli can do what we call fermentation, right? So it's a very, it's abundant organism in our guts, it's abundant in the soils and sediments. So it's got a wide variety of choices when it turns to ways to make a living. C. diff is in this family of what we call Sticklin fermenters, where you have a variety of what we call electron donors. These could be sugars. These could be aliphatic amino acids like leucine and isoleucine, valine. And they could even be hydrogen gas, although that's been pretty poorly studied as far as C. diff is concerned. As long as you can oxidize this metabolite or carbon source and generate generate reducing potential, which is NADH generally, you have to have an electron acceptor to balance out the reaction. So what's interesting about Sticklin reactions is, and especially in C. diff, proline turns out to be the preferred electron acceptor, and it's not fermentation, although it's mislabeled in many, many publications and biochemistry textbooks. Proline is actually respiration. So E. coli is basically, C. difficile is basically breathing proline because it produces delta aminovaleric acid, and that's just an end product. And there's also evidence that this enzyme is tied to what we call proton motive force in the membrane of the C. diff as well. Not absolute molecular mechanism has been described yet. Now the enzyme that Terry started studying, glycine reductase, this mechanism of this enzyme is actually well established by Terry and another group to be substrate level phosphorylation production of ATP. So classic cellular bioenergetics as well. But C. diff seems to prefer one of these two enzymes when it's growing in culture. This is some of the earliest work on donors and acceptors. It's now established that there's lots of different donors and acceptors in various types of Clostridia. And glycine, proline are some of the highest acceptors that were identified early on. So what my graduate student Sarah Jackson did was she started growing C. diff in the lab a number of years ago and asking whether growth would be increased when we added donors and acceptors and selenium. And she found that in the presence of the right donor, acceptor, and sodium selenite, like what Bob Lester had added to E. coli, we got the optimal growth of the bug. We showed that the genes encoded the selenoproteins. This is actually what we call a radio-labeled gel looking at incorporation of selenium into particular proteins, so we can actually see glycine reductase, proline reductase in these gels. They will turn on glycine reductase in the presence of glycine and proline reductase in the presence of proline. The mechanism by which they do that is now recently been established in terms of transcriptional regulation. So C. diff is a Sticklin fermenter, we showed that, or a Sticklin organism. So I could add even more bullet points to this today. A number of studies looking at animal studies, especially microbiome studies, have now shown that Clostridium difficile upregulates very highly proline reductase in the gut of the animals. And it seems like, and even recently a lot of papers have been published that show proline is the preferred electron acceptor in animal models, et cetera. There's actually a recent paper, which I don't have on here, which showed that if you took away the ability to do proline respiration, then you mitigated the damage in an animal model as well. So proline seems to be preferred, glycine as an alternative. Is there a link between Sticklin fermentation and pathogenesis? Pathogenesis being mediated by toxins. So this is a Nature Review slide. Toxins mediate the disease in C. diff. That's really all you need to know. And so if you affect toxin production, you're going to affect the disease. And so what is the relationship between central metabolism and toxin production in C. difficile? and we still really don't know. So this is one of Bruno Dupuis' figures. Antibiotics, different antibiotics will trigger toxin production or suppression. Temperature can do it, carbon sources. Many years ago, glucose was shown to suppress toxin production. Amino acids, butyrate has also been shown to be a mediator of toxin production. This is an end product of fermentation. The genes and the regulation of toxin are pretty well described now, and actually Shona McBride published a paper about a month ago that described a two-component system regulating toxin production. This is very, very recent work out of a collaborator, Ling Sonnenschein's lab with Laurent Boulet. And here is an updated sort of scheme of the basic core bioenergetic pathways in C. difficile. So we have a part of what you will learn or have learned as classic glycolysis pathways here, glucose oxidation to pyruvate, generation of NADH. This is the proline reductase complex. This is basically generalized amino acid oxidases that can generate NADH, glycine reductase. There is an alternative Stickland reaction here that it mediate through the CoA, what we call CoA-dependent enzymes, crotonil, butyryl-CoA. So you have these classic Stickland reactions, you have these other Stickland reactions, all contributing to C. difficile growth in the host, probably. This is, of course, in vitro. This is this paper was just published, actually last month about this time. What's really interesting is when you take the ability of the organism to upregulate proline reductase, right, its preferred electron acceptor, take this in a PRD mutant, this is actually showing, this is a far higher toxin producer in the animal. So these animals are much sicker, much faster than the control animals in these cases. So this shows clearly the decisions on how to make a living, the decisions on how to make ATP at the substrate level or at respiration, and the decision to make these big expensive toxins are somehow tied together. And yet we're still trying to learn exactly how that occurs. So this is some unpublished data that my student Sarah Jackson had done a number of years ago that we're following up on now. We grew C. diff strain that produced a lot of toxin in vitro in the presence or absence of proline, glycine, and hydroxyproline, which is something you find in collagen, derived from collagen. The growth was about the same. This is what we call rich medium, so these organisms aren't starved for nutrients. This is really asking the question, if you grow C. diff in a medium where it's growing well, and you throw a lot of each of these amino acids at it, what does it do to toxin production? And so it turns out that the addition of glycine ramps up toxin production pretty robustly in this case. Proline, with respect to the control, is not so much. Glycine, we maybe understand based on some of the regulation that's been published recently, but hydroxyproline, which also benefits growth, which also we now know can be converted to proline, completely suppress toxin production. And so these are sort of these really cool observations which are looking for a molecular mechanism. So that's what we're going after in the future here. The tRNA, I should say the messenger RNA levels completely match the levels of protein that we identified. That's not a Western blot, that's a large Clostridial cytotoxins that have been running a really low percentage STS page gel. That's like a 4% gel. And you can identify them directly. This is a TCD8 toxin right there. So we know it's at the transcriptional level because the mRNA matches it. And when we looked at toxin production genes, we also see the same kind of regulation. So we're now going back to really look at this data in the light of some of the published papers that have come out in the last couple of months to figure out if part of the mechanism has been established and what part hasn't. So again, there's a lot that's come out, there's a lot of new information, and we're trying to figure out exactly why C. diff makes those decisions. And as I mentioned, what exactly is the role in the ecology of C. diff in the host? Why do they produce the toxin? What do they get from the toxin? Why is it correlated with bioenergetics as well? So I think I'll probably just show you a couple of slides of some other work that Sarah did in the lab, and it was based on an accident, like everything else. So typically, you don't intend to do it. This was a different Sarah. So this was Sarah Mino. She was doing a study with arsenic and looking at how arsenic affects selenium trafficking in human cells. And she ran a control with a compound called aranophen, which is a gold-containing drug. And she actually showed that Aranophen actually blocks selenium trafficking in human cells. So when I was talking with Sarah Mino when she was working on this, and I told the other Sarah, I said, well, you should try this with C. diff, because if it blocks selenium trafficking in C. diff, it should block growth, because we think Stickland reactions or selenium-dependent Stickland reactions are absolutely required for growth. Of course, kind of think they are, we kind of think they might not be now. So basically, this is what she got. So this gold-containing drug, which has been now studied for cancer research. It's an old rheumatoid arthritis drug that's still in the formulary, but it's not hardly ever used at all. Shown to potently inhibit growth of C. difficile, no matter what strain you throw at it, with a very, very tight MIC, 1 to 4 micrograms per ml, which is pretty good as an antimicrobial. We showed that it didn't inhibit growth of other Clostridia that don't make selenoproteins, and if you added selenium back to the culture medium, it overcame the effect, which tells you that you're hitting something as a nutrient. If it was hitting, the ribosome, you wouldn't be able to overcome the effect on the ribosome by simply adding selenium. So this sort of hinted at strongly the molecular mechanism. We did it in E. coli, and we were able to reduce the incorporation of selenium in E. coli, but E. coli doesn't need the selenoprotein, so it doesn't inhibit growth to any great extent. This is actually an E. coli gel where you can see the formate dehydrogenase that Bob Lester was enhancing by adding selenite. You also see the modification of tRNA, which I didn't talk about, which is a whole different use of selenium, which is still, it's well established how it happens, but what exactly it does for the ribosome is still pretty poorly understood. You can see in C. diff, it really almost completely suppresses incorporation of selenium. This is actually incorporation of radioactive sulfur as a control, just showing that the ribosome still works. We're still incorporating sulfur into proteins, but we're not incorporating selenium. And we actually showed that the chemical complex that's formed between the drug and selenium, and part of this analysis was done by a friend of mine, Ron Devine, a number of years ago. So we delved into the antimicrobial business. The fun part about that is that the report many years before we did it showed that this compound actually could inhibit growth of about 30 different bacteria. So when we went for a patent, then all the intellectual property was lost many, many years ago. So no matter how good a compound it is, without any IP, we couldn't get any IP from the patent office. Nobody can get any IP. It's sort of one of those lost drugs that can't be used, kind of a sad part of the patent law. So there's an acknowledgement. Some of the more recent students, especially Daniel Murdoch's been doing some growth studies in C. diff, and a number of students in my lab working on other projects. So that's sort of a synopsis on some aspects of how C. diff makes a living, and I'd certainly take any questions from you guys. Question?
You had a couple of, way back, you had a couple of pictures of colon and everything. Could you explain what they were?
Pictures of what?
Of the colon, inside the colon and kind of
Oh,
outside the colon?
yeah. Well, no, probably not, since I'm not a physician, but sure.
It was way back towards the beginning.
Yeah. So that's, so one of the,
There.
this is what they call pseudo membranes, right? So pseudo membranes are produced in the colon in response to C. diff toxin. I mean, and I don't follow that literature that tightly, but I still don't think we know why they make tons of membranes. In other words, the colonic epithelial cells just start producing lots of membranes, and that's sort of the mechanism behind this yellowish stuff. But why the toxins trigger the membrane production is still pretty poorly understood. Again, why they make the toxin if they're not trying to invade. I mean, what I didn't tell you is the analogy is botulinum toxin, right? So botulinum toxin is a neurotoxin, so it's very clear what its mechanism is. It's to get into a mammal, mobilize the brain, and kill the animal. And so, and from a group perspective, Clostridia eat you from the insides out, because we all are full of Clostridia and anaerobes at any given time. And if we happen to die, well, then they're going to literally eat us from the inside out, because they're good at breaking down collagen, and we're full of collagen. And, sort of the natural decomposition of mammals is, the anaerobes are going to eat you from the inside out. There's actually a place in Tennessee called the Body Farm Research Lab, where they study how anaerobes eat you from the insides out when you're dead. So, but botulinum clearly has that goal, because it's gonna, and tetanus too, because tetanus causes lockjaw, tetanus causes you heart to seize up and for you to die. But C. diff doesn't do those things. And so the biological rationale for C. diff toxins is still, I would say, not understood. Not to say it doesn't cause a lot of disease, but the point is that, you know, these organisms, they produce everything for a very good reason. Bacteria are super efficient. So
The other picture? No.
that is a form of what is called toxic megacolon. I don't think that living patient, I'm not sure. But that's when it's progressed typically to where it's fatal. Yes?
Do you need both toxin A and B, or can it be one or the other?
Good question. So there's a lot of argument in the field about that, especially early on. So the genetics in C. diff, you know, when I started at UCF 16 years ago, were almost non-existent. They basically were non-existent. When I say genetics, the ability to delete something or remove the gene and look at that. Some of the earliest papers, I think the first paper with a knockout was one of the toxins. It was toxin A. And so you can identify toxin B containing toxin A minus strains or toxin A containing toxin B minus strains. The toxins have been pretty well described in how they are taken up by cells and how they're matured. And I haven't seen a lot of work since those early papers that has argued that one isn't more important than the other. Most strains have both. So it's not as if there's a wide variety that 10% of strains are toxin B minus and 20% of strains. It's pretty uniform that toxin A and B are almost always present in both. And that's pretty remarkable because an earlier paper with the title something with respect to a mosaic genome, the rest of the genes, or I should say a lot of the genes in C. diff are expendable. If you look and you isolate 100 strains of C. diff from patients or from sources all around the world, and you say what's always there or almost always there, you'll find genes like the toxins, you'll find proline reductase, you'll find glycine reductase, you'll find some of the core metabolism, of course ribosome, all the stuff that you can't get rid of. But toxins and the core metabolic genes have been identified in a number of strains. One thing that a toxin minus strain that doesn't make any toxin, how is it making gluten, right? Because is it causing an infection? Is it going along for the ride? I don't know. Does it have another host? Those kind of open questions. Yeah.
The toxins are in all strains, but was that of the hospital strains that are causing diseases, or what about the non-disease patient?
Right. So.
And what about the soil?
Soil,
that wouldn't have it, would it?
I would love to see. Well, almost nobody studies the soil because they can't get money to do that. Because most of the strains are going to be from the clinic. The vast majority of strains that have been sequenced or isolated are going to come from human clinical samples. So that probably biases it, right? So if you actually did a really good ecological study and went and identified 100 strains of C. diff everywhere you could find them, would you find a lot more toxin minus strains? Probably. But then you'd also look at what would correlate that's present, say, in the sediments or present in the soils. That might be what they're doing to make a living in that environment. So they're not on some kind of plasmid or some what they call pathogenicity island like Salmonella has, where it migrates, something like that. So, and they're also a family called what they call large Clostridium cytotoxins. So there's a number of strains I haven't even talked about that produce toxins very much like this, but don't invade the gut or don't have effects in the gut. Sordelii is one of those. A question?
You keep referring to it as making a living, and I was just wondering, is there any talk amongst the scientific community of like conspiracy theory and how this all began, and potentially like pharmaceutical companies
Right.
trying to pre-plan the moneymaker or
No,
population
probably.
control, none of that?
No, I don't think so. These organisms have been around for a long time, probably infecting mammals for, I don't know, millions of years.
But to put it as an additive in food, and then
Well, now that
posed with a lot of, the right
Right.
ingredients for breeding,
Yeah.
so to speak. I mean, they seem kind of thought out.
Yeah, I get you, but no. So the trehalose, from what I can tell from Rob Britton's paper, the trehalose thing was something they look like they accidentally discovered too, because they actually used a system where you feed your organism of interest, like E. coli, C. diff, all the different kinds of carbon sources you could ever find, and all the different nitrogen sources, called a biolog system. And so they did a biolog experiment where I think they were surprised to see, well, look, it grows really well on trehalose. And it's only this set of strains that grows really well on trehalose. Those set of strains have been around for decades before that. It wasn't like they just came out in 2001. So what they did notice is that that set of strains that happens to grow good on trehalose expanded after 2001. So that was really the discovery, because those strains have been around and cataloged decades before. And so, yeah. And it was an accident. Yep.
What is the goal of all this research? Like you say, we're gonna cure cancer, we're gonna.
Right.
Okay, you've done all this work, they're in process of doing all this work. What are we trying to solve?
So the goal of the Aranophen was an accidental thing. What we pursued on that was drug discovery, because we had noticed that it was pretty good at inhibiting growth of C. diff in vitro. But again, we hit the wall in terms of intellectual property on that one. We actually have screened a lot of marine natural products with a collaboration with a scientist at Harbor Branch down in Fort Pierce. And we actually have a compound that we've identified from a marine sponge, which has pretty comparable antimicrobial activity against C. difficile. Mechanism probably completely different, but we're pursuing, we have what they call a provisional patent on that. So we're in both ends of this sort of like game. One is to understand the basic biology of the organism so that we can understand it better. Maybe that could lead to our ability to then target those pathways if we find them to be absolutely essential. And then the other is sort of applied research, where we're just looking for new novel compounds that could inhibit growth of C. diff. The majority of drugs on the market today that are used to treat C. difficile are natural products, or derived from natural products that were done in sort of screening platforms to find inhibitors of growth. So that sort of classic drug discovery, go find something that inhibits. The ideal compound to treat C. diff would be something that is, and that's where Aranophen was pretty exciting, because it didn't affect E. coli, it didn't affect other bacteria that we tested, at least. Of course, I'm sure it would affect other bacteria. But it was poorly absorbed, so you don't have to worry about systemic, it's in the gut. And you would want something poorly absorbed, relatively specific to C. diff, that has very high potent ability to kill the organism quickly. And so the most recent compound that came out probably about 5 or 6 years ago is actually an RNA polymerase inhibitor that is not very specific, but it's very, very good at hitting RNA polymerase, and at the same time it prevents toxin production, because you're not going to make any more mRNA. So it was shown to be pretty good. What I didn't talk about at all, which is another relevant question, is recurrence. So it turns out about 25%, 20 to 30% of patients who get a C. diff infection have a recurring infection later. And we really still don't understand that. Is it because, and we really, I would say that, the scientific community doesn't think it's because they never got rid of it. It's probably because the spores of that organism are either all over the hospital, or all over their house, or wherever they were when they got it the first time. And then they're still susceptible to it, because it's not like a cold where you get an immune response that says we're not going to have a C. diff infection again. So recurrence is still a real problem in the community, that only new compounds maybe can help. Does that help to answer your question?
Yes.
Yeah.
So on top of that, how long is it viable to be outside the body, and
Oh,
is
yeah.
temperature related? Because I know some pathogens aren't temperature related outside of the body.
Yeah, the spores are really long-lived. I mean, they're hundreds of years a spore could live. So they're desiccant resistant, so they can be dry, hot. Once C. diff spores are made, and they sporulate a lot of spores when they're growing, they're tough. Bleach will get them, but what I've heard from my friends in the clinic, they don't like to use bleach a lot. And alcohol, it's not so good. So the hand sanitizers, there's a lot of controversy in the nursing community over the alcohol-based hand sanitizers where they work on C. diff spores. So they're tough, and they make a lot of spores. So, or humans make a lot of spores when they have a C. diff infection. And you probably, if you worked in the clinic, can smell it. We can identify it by smell.
See, I don't have that ability, I can't tell.
So you're lucky.
Oh, yeah, oh, yeah.
All right.
Very much.
Thank you.